Good morning.
Good morning.
Good morning.
Welcome, everybody. Happy Monday. If you would, go ahead and type your first name into chat for me. Thank you. Very good. Thank you. We're still waiting on a couple people, but we're just at 11:20, so I'm going to just go ahead and begin. Just a reminder to everybody that you have a lecture exam today. A couple of you have already taken it, which is great. You're just going to want to take it at some point during the day today, whenever your schedule permits, and get that submitted by 8 p.m. If for some reason that's not going to be possible, contact me as soon as you can after our meeting today. And if needed, we can extend your due date time if you're working or something and you need an extension on the time this evening. That's no problem. All right, very good. A couple more folks in. You also have a lecture topic this week. You only have one lecture topic this week because of the exam. And that topic is the prokaryotic cells. And a quiz goes along with that. And of course, for lab this week, we're going to be talking about something called Aseptic Technique. We'll do that today, or at least we'll get most of the way through that today. And then we're also going to be talking about a differential staining procedure this week. Very, very commonly used differential staining procedure called gram staining. So that'll be coming up this week as well. And of course, you'll have homework to do regarding the lab material, as usual. Just a reminder, next week coming up, a week from today, you have your first laboratory practical exam. So basically you're another exam that's based on laboratory material only. And we'll talk more about the details of the lab practical exam later in the week, but just so you have that in your head, that's coming up next week. This week is the time then, if you need to, if you missed any of the labs and you need to watch a recording of a lab you missed, or if you want to go back through your notes, your lab notes, and clean up a few areas, this is the time to do it this week. Because of course you can have your lab notes next to you while you take the lab practical exam. So just a heads up about that for next week. Oh, and I also wanted to mention that most of you have been doing this and I really appreciate it. But if you need to miss a lab, please let me know. Please send me an email and let me know. Preferably before the lab, but I understand that sometimes things happen very suddenly and you can't email me before the lab. But if that happens, just shoot me an email when you can and let me know why you weren't in the laboratory. These sessions that we're together on Zoom are essentially the same as if we were in the classroom, and so your attendance is required at those. But I certainly recognize that things happen from time to time and you just can't manage it for one reason or another. And that's part of the reason that I post recordings of our labs, so that if you have to miss a lab from time to time, you can go back and watch that recording. All right, very good. Just about everybody's here. I think we're only missing a couple of people. If you haven't already, type your first name into chat. That's my recording of who was here. if you come in late to a lab, if you can remember to type your name into chat, all right? Sometimes I recognize sometimes you just are a little tardy getting started. That's fine. Just make sure to remember to type your name in so that I have a recording that you were here. All right, first things first, does anybody have any questions about anything you've been reading or watching? Any of the quizzes you've been taking, anything at all? I know you have an exam today, so hopefully you're feeling good about the material that's covered on the exam today. But if anybody has any questions they'd like to ask, you can feel free to do that. You can type your question into the chat, or you can unmute yourself and call out.
Can you please go over question number 4 from the lecture quiz, The Science of Microscopy?
Microscopy? Yeah.
Please, yeah.
Do you remember the question at all?
I don't remember the question. I'm
Do you remember
sorry.
anything about it, like what the answers might have been, or just to jog my memory?
No, I remember. I just had like a comment on my quiz, and you said that you'd be willing to go over number 4, but
Okay.
because I had gotten it wrong.
And that's perfectly fine. Give me just a sec here, and I'm gonna pull up. I'll pull that quiz up and we can take a look. Okay. I'm gonna share my screen with you. All right, lots on my screen. You should see, now you should see our Blackboard page, the home page. So I'm just gonna go into course content here. I'm scrolling down into our second week and into our quiz. Okay. There were a couple questions on this quiz that were a little bit tricky. Okay, so question 4 was about this resolving power property that microscopes have. It says light microscopes have a resolving power of about 200 nanometers. If two objects on a slide are located 100 nanometers apart from each other, will they appear as two objects or will they appear as one? Okay, and again, many of you got this one right, but this is a difficult concept. So let's talk about this again. Resolving power is a number, a quantity that tells us how far apart two objects need to be on the microscope slide in order for the microscope to be able to show it to us, magnify it enough for our eyes so that we can see it as two separate objects. All right, if you think about it, when things get magnified, there's a limit to how magnified they can be under a microscope, under a light microscope. Let me put it that way, because an electron microscope has a very different resolving power. So most light microscopes actually have a resolving power of about 200 nanometers. And what that number should mean to you is when you see that 200 nanometers, you should be thinking that means that two objects that are sitting on that slide, two cells or whatever, need to be at least 200 nanometers apart from each other in order for us to see them as two different objects. In other words, if they're closer together than 200 nanometers, the microscope can't resolve them enough. It can't separate them enough for us to see them as two. So we're going to see them as one. So anything less than 200 nanometers apart, and our eyes are going to see it as one thing. So the correct answer for that would have been 1. The two objects were 100 nanometers apart, and the resolving power says they have to be at least 200 nanometers apart for us to see them as two objects. Again, most light microscopes are right in that 200 nanometer range. Remember though, that's still pretty close together. Remember how small a nanometer is. You know, there are 1,000 nanometers in 1 micrometer, and there are 1,000 micrometers in 1 millimeter. So 200 nanometers apart, those two things are still pretty close together. Does that make sense? Do you understand what resolving power is about?
Yes, thank you.
You're welcome. So the thing you need to remember is that it's anything less than the resolving power, you can't see them as two different things. The distance between two objects, if it's less than the resolving power, you're not going to be able to see them as separate things. You're just going to see them as one thing. If it's greater than or equal to the resolving power, you'll be able to see them as two different objects. Good question. All right. All right, well, I'm going to go ahead now and go into our slides for the day for this lab that's called Aseptic Technique. Today, we're going to be talking about, again, some sort of basic information about how we handle ourselves in the microbiology lab, because we're going to begin talking now, including later this week, about actual procedures that we do. We're going to start learning about staining procedures in more depth, and we're going to talk about different diagnostic testing that we do in the lab and procedures that we use to place microbes onto or into culture media. So we need to talk about the technique, just the general technique that we use while we're in the microscopy lab, microbiology lab. I have microscopes on the brain. In order to be successful in our work. We're also going to talk a little bit today about some of the things we mentioned in the biosafety lecture about things like waste management, because that kind of falls into this idea that we're always thinking about techniques related to asepsis. In other words, we want to do things so that we do not contaminate. So the first thing we're going to do today is define what we mean by this term aseptic technique. And then we're going to spend our time together talking about why these techniques are used in the laboratory and also how we implement these techniques. So we need a definition. What do I mean when I talk about aseptic technique? When I say that somebody has very good aseptic technique while they're working, or perhaps somebody has very poor aseptic technique while they're working in the laboratory. What I'm talking about is all of the methods that that person is using in the laboratory to prevent contamination. And remember our golden rule. We want to prevent ourselves from getting contaminated, and we also want to prevent our cultures from getting contaminated. We don't want to put ourselves in danger or put anyone else in danger by contaminating ourselves with these cultures that we're using, that we're making in the laboratory. We also don't want to contaminate our materials with ourselves. We don't want to get microbes that are on and in our body into our cultures that we're making or our diagnostic samples that we're handling. So that's what all these techniques are geared to do. Prevent both kinds of contamination. Now, there's a couple important things to just remember, and that's number one, microbes range from harmless to pathogenic, right? We've talked about this at length. The vast majority of microbes that we encounter in the world, whether in or on our bodies or out in the environment, are either harmless or actually helpful to us. Remember, it's like 95% of all the microbes that we bump into every day are completely harmless to us or even helpful to us. We need them to be healthy. But there are pathogens, up to 5% are pathogenic. So that means we have to treat all of pure microbial cultures in the laboratory as though they were pathogens. If we learn how to do that, if we learn how to treat every single culture, every petri dish, every test tube that has microbes growing in it, that's what we mean when we say culture. If we do that, if we treat them as though they were pathogens, then if the time comes when they really are pathogens, we'll be fine. We'll be good because we treat everything as though it was a pathogen. Now, I want to mention a couple words about culture media because that applies to aseptic technique in an important way. So when I use the word culture, I'm talking about microbes that are purposefully growing in some kind of material, some kind of media. Remember, some of the culture media is solid, some of it is semi-solid, some of it is liquid. We use all different forms of culture media. But culture media is made, it's designed to have nutrients and things in it that microbes need in order to grow, in order to divide and increase in number. So culture media is designed to support growth. And while we put the microbes that we want to grow into the culture media, if any contaminating microbes get in there, they're going to grow too. So that's why we have to be so careful. We are creating these solids and liquids that are just perfect for growing microbes. So if any contaminants, any microbes fall out of the air or get off of our fingers or out of our breath into our culture tubes, those are going to grow too. So we inoculate our culture media purposefully. We put the microbes into the culture media that we're trying to grow. And we put that culture media into some type of incubation. Now, there is something called an incubator, which is a cabinet, essentially, that is kept dark and warm. So a lot of microbes need a warm temperature in order to grow optimally. So we put our culture that has been inoculated with cells, we put it into an incubator, again, to give it the conditions it needs to grow. But even though you may hear me use the word incubation, that doesn't always mean that we are putting it into an incubator. That's an important thing to put down in your notes. It is possible for some microbes to grow perfectly well at room temperature. So sometimes we, quote, incubate, unquote, cultures by just putting them on a countertop. Most microbes, in fact, all microbes, need to at least be in the dark, or I'll say in the shade. But temperature requirements really vary. So sometimes we incubate on the countertop. Sometimes we incubate in an incubator that we have warmed up. If you think about it, if you're a bacterium, for example, that grows in the human body, you're accustomed to human body temperature. So we need to get the incubator approximately to human body temperature in order for you to grow and be comfortable and make new cells. Growth media is designed for these cells to divide in. They love it. It has all their nutrients. It has everything they need. So what we put in there is going to grow, but any unintended contaminants are also going to grow. We've got to handle our media very carefully to prevent accidental contamination. This is a busy slide. It has a lot of information on it, but these are the general rules for handling culture media. Remember, when we start, right before we inoculate our culture media with microbes, it's a sterile material. So, number one, we never leave an agar plate or a broth tube or any other type of culture media container. We never leave it open any longer than absolutely necessary. Obviously, we have to open it because we have to put our microbial cells into it. But we want to do that as quickly as possible. We want to open it, we want to do the transfer, we want to close it. Because as soon as we close it, we're preventing any more accidental contamination. Number two, we have to always assume that the surfaces around us are contaminated. We always assume that. So even if I just disinfected my workspace with alcohol, I'm going to assume it's contaminated. So that means I will never take a sterile lid for a Petri dish, or I'll never take a sterile cap from a test tube and place it down onto a surface. I'm always going to keep that lid or that cap or any other sterile tool I'm using in my hands. Because if I place it down, I have to assume it's going to get contaminated. Remember, microbes fall, they don't fly up. So that means just opening a tube doesn't mean the microbes are going to come out. Just opening a dish doesn't mean that the microbes are going to fly out. They're not. But it does mean when I open those containers that things that I don't want in there can fall in. And finally, the last thing on the slide says, nearly all forms of contamination are carried on dust particles in the air, including microscopic dust particles in air currents. If you're working in a laboratory space, there's a ventilation system in that room. There's a ventilation system in the building, and it blows air. So you're going to have air currents in the lab, and those air currents are going to have little microscopic dust particles in them. And the microbial cells and particles, like viral particles, are going to be sitting on that dust. So the longer we leave a container open, the more likely it is that some of that dust is going to fall and land in our container. So we just have to always be thinking about that. Now, I said at the beginning of our time together today that I wanted to touch on a few things about waste disposal. And this is a good time to do it, primarily because we're talking about culture media. So I've got, again, a lot of information on this slide, but this is the best place for us to sort of tuck it in to laboratory material. We've said already in here that there's chemicals that we use in the microbiology lab that cannot go down the drain. So one of the ways that we dispose of chemicals is by what we call hazardous waste containers. These are just containers, but they are specifically labeled so that we know that they contain certain hazardous chemicals. So all of our stains go into a hazardous waste container, and many of our other chemicals have to go in there as well. And those containers are disposed of very carefully. They're picked up by professional commercial waste disposal companies so they can be decontaminated and disposed of safely. Of course, we also have regular trash in the lab. And a lot of things go into the regular trash can and they just get thrown out. Things like our used gloves, paper towels that we may have used on our desktop, Kim wipes that we may have used, lens paper that we may have used to clean off the microscope lenses. All of those kinds of things just go into the regular trash can. Students sometimes get confused because they say, well, we're supposed to imagine that our gloves are contaminated. So how can we just toss them in the trash? And the answer is, even if your gloves are contaminated, even if you accidentally got some culture, some living microbial cells onto your gloves, those microbial cells are not going to be able to survive on those gloves. They need a certain things. They need water and they need nutrients and so on. So just having a couple cells on the glove, when you toss the glove into the trash, those cells are going to die pretty quickly. Now, I have a couple things on this slide that have a little asterisk next to them, and those are the sharps containers. You've probably heard of sharps containers. You've all seen them if you've ever been in a doctor's office or a hospital. These are the red boxes that are used to dispose of things like needles, hypodermic needles, and syringes, and so on. So the thing to know about the microbiology lab is that there are typically two different sharps containers in the room. There's what we would call just a plain old sharps container, and there's what we refer to as the contaminated sharps container. Again, this might seem strange because in a doctor's office or a hospital, all of the sharps containers are contaminated sharps containers. They all contain things that have touched patient body fluids. So, you know, when you use a syringe with a needle on it to draw blood from a patient, when you're finished, you put that needle into the contaminated sharps container because it's been contaminated with that patient's blood. It's hazardous, it's biohazardous. But in a micro lab, we keep another sharps container available that is not for contaminated materials, things that have been contaminated with microbes. We have another one that is just for sharp things. I'll talk more about these two types of containers in just a minute. We always have a glass waste disposal container in the laboratory that is designed for microscope slides, primarily cover slips, and any other accidentally broken glass. Now you'll notice I say clean broken glass. If I had a test tube that was full of culture media, let's say it had broth in it, and I had grown microbes in there. So I had a nice microbial culture in that tube. And let's say I accidentally dropped that tube. There are procedures to follow to clean up that culture that has dropped on the floor. But the glass, since it had culture growing in it, that broken glass has to be autoclaved. It has to be sterilized before it's disposed of. But most glass in the laboratory doesn't have to be autoclaved. It doesn't have to be sterilized. We just have to dispose of it into a special container because glass is sharp. It tends to break and it has sharp edges. So we want to make sure that all of our glass is in a container that when the janitorial staff comes to take it away, we're not risking that they're going to get cut. And finally, we have the long list of things that go into the autoclave bag, or what we sometimes refer to as biohazardous waste. I'll show you a picture of one of these bags in just a minute, but all of the things that we use in the microbiology lab that are actual culture containers and things that come in direct contact with those cultures, those things have to be sterilized before they're disposed of because we don't want to put our microbes out into the environment where they could potentially harm someone. So all of our Petri dish plates that we grow microbes on, all of our broth test tubes that we grow microbes in, those things have to be sterilized before they're disposed of. If we were using an inoculating loop that was disposable, and we haven't seen one of those yet, but if we were using one of those, we would, because it touches the culture, and because it's a single use item, we would want to autoclave that before we threw it away. So the general rule is things that come into direct contact with our pure cultures and our pure cultures themselves get sterilized in the autoclave before we throw them away. And we have a special bag that we put all those things into so that they can go into the autoclave. And I'll show you again what those bags look like. I do have a note at the bottom that we always have to remember that there are some containers that we use in the laboratory that are reusable. And that's a good thing. That's a good thing because we all need to reduce the amount of waste that we generate, including in the laboratory. So we do use glass test tubes and flasks and things like that. So if I grow a culture in a plastic test tube, when I'm finished with that culture container, I'm going to put it in the autoclave bag so it can be sterilized before it gets thrown out. But if I'm using a glass test tube and I've put broth into it and I'm going to grow microbes in it, I'm going to want to keep that glass test tube. So what do I do there? Well, you empty the culture material. You empty the broth with the cells growing in it into the autoclave bag. But then you take that empty glass test tube that's contaminated and we wash it and then we put it into the autoclave. What will happen is it will be sterilized empty in the autoclave and we'll be able to use it again. So some things that we're putting into the autoclave to be sterilized are things that are going to get thrown out. But other things that we put in are things that we're sterilizing so we can use them again. In hospitals, of course, the majority of what goes into the autoclave in a hospital setting, if you think, for example, about the operating room, all of the instruments that get used during a surgical procedure, those aren't single-use items. Those are metal, very expensive instruments. So they get used during surgery and contaminated with body fluid. And then they are washed and then they are packaged and put into the autoclave so they can be sterilized and used again. Surgical drapes and surgical gowns, those are all autoclaved in order to be sterilized so they can be used again. Although some of them now are single-use throwaway items. So just a note to remember that some of the containers that we use in the microbiology lab are reusable. Unfortunately, a lot are single-use plastic, and they just get thrown away. They get put into the autoclave bag, they get sterilized, and they get disposed of. All right. Anybody have any questions so far? Any questions about what I've said so far that you might like to comment on? I get a question a lot from students about what do we do with the slides that we make a smear on and then stain? When we're finished with that slide, where does that slide go? It seems like that slide should have to go in the autoclave, right? Because I just said anything that comes in direct contact with pure culture should be put in the autoclave and sterilized before it's thrown away. This is kind of an exception to that though, right? Because if you remember, we have heat fixed that smear and we've stained it. So that means that all those microbial cells are dead. So when we finish with a smear that has been stained with a glass slide that has a stained smear, that one can just go in the plain old glass waste container. We want to put it in the glass waste because microscope slides are made out of glass and they tend to be sharp, they tend to break. So it has to go into that for the physical hazard. But we don't have to sterilize it because we've already killed all those cells by heat fixing it and staining it. And as I said, you might also think, well, my gloves should go into the autoclave bag because, you know, I'm supposed to think of them as being contaminated. But remember, most of the time your gloves aren't contaminated. If you're doing what you should be doing in the lab and you haven't made any errors, your gloves aren't contaminated. But even if they do get contaminated, the number of cells is so low and the cells survive for such a short period of time on that material that we're safe throwing it in the trash. All right. Lots of things to remember when you're working in a lab. All right, I want to switch gears now and talk about these techniques. You've actually learned some of them so far. Remember, when we talk about aseptic technique, it's actually a group of things that we do. There's not one aseptic technique. It's a whole group of things that we do in the lab. So you've learned a couple of these already. You've learned that we should always decontaminate our workspace. We always want to disinfect our workspace with either 60 to 90% alcohol or 10% bleach. You know that you're not supposed to keep any of your personal items in your workspace. You know that if you're going to use a metal inoculating loop with a wire on it, that you have to sterilize that either with an open flame or with an incinerator. We're actually going to show that today. We're going to look at what that looks like when it's done. We're going to look at how it's the wire portion of the inoculating loop that we have to sterilize. And we're going to show how we know that we're at a hot enough temperature. That's red hot, literally. And we're also going to talk about how after we sterilize our metal inoculating loop, we have to let it cool. It takes about 20 seconds for a loop to cool after it's been sterilized. So we don't want to stick our loop into our culture container until it's cooled off, because a hot loop will kill the cells that we've been trying to grow. So the new techniques that we'll talk about today are listed on this slide. We'll talk about how you flame, as we call it, flame a glass test tube, how we transfer bacteria from one tube to another. In other words, how we would take some cells out of a broth culture tube and put them into a new sterile broth tube. We'll talk about how we transfer bacteria from a broth test tube culture onto an agar plate. We'll talk about how we handle agar plates properly. We'll look at a couple of other tools that are very commonly used in the microbiology lab. And again, I'll show you the autoclave bag, what it looks like, and how we handle it. So I do want to say a couple more words about these sharps containers. You've all seen these red boxes if you've ever been in a laboratory, if you've ever been in a doctor's office, or you've been in a hospital setting where you've been treated. These are sharps containers. These are commercially available containers. They're made out of hard plastic. And these are intended to keep not only sharp items out of the regular trash, but contaminated items out of the regular trash. And here, when I use the word contaminated, it could be any kind of contaminated material. So it could be body fluid, or it could be a culture that we made purposefully in the lab. Now, I want you to look at this sticker here that's on this box. It's hard to see, but there's a biohazard symbol right here. This is a universal symbol that tells people that the things that are in this box have a biohazard associated with them. They are biologically hazardous. So again, it might be because they've touched blood, or they've touched urine, or they've touched tissues, or they've touched a pure culture in the microlab. So in any microlab, we're going to have these sharps containers that have the biohazard symbol on them. Here it is enlarged. You can get these boxes in all different sizes and shapes too, which is good. So this is what that symbol looks like right here. This is universal. So lots of things that we handle in the microbiology lab are not only sharp, but they're contaminated because they've touched culture. Once they're full, once we have filled up one of these boxes, it has to be handled just like biological waste. It has to be, again, autoclaved in order to sterilize it and then thrown away. But in some laboratories, including the micro lab, there's going to be a second sharps container. And that's why I want to mention this. There's going to be a sharps container that doesn't have that biohazard symbol on it. It's just a hard plastic container that's intended for sharp things to be disposed of. But these are things that have not been contaminated with body fluids or with microbial cultures. Now, you might ask yourself, why in the world would you do that? Why would you have two different sharps containers? Why not just throw everything you have into that one biohazard labeled contaminated sharps? Why in the world would you bother to use two? The answer is all about, sorry, here we go. The answer is all about money. Of course it's about money, right? These are expensive to maintain in the lab because they have to be handled and disposed of as biological material. So if it's labeled with that biohazard symbol on it, we assume that the sharp things in here are not only sharp and dangerous to us, but they're also contaminated with microbes and dangerous to us. And again, when this thing is all full, we either have to autoclave it and dispose of it, or we have to send it along with a commercial company who comes and picks it up and takes it away to be autoclaved and thrown away. It costs more money to handle sharps in a biohazard way, a biohazardous way. So in a lot of labs, you're going to see this, including microbiology labs. This is, for example, an old liquid laundry detergent bottle. This is an old, I don't know, a coffee container or something. A sharps container by law has to be hard plastic. It has to be plastic that you cannot see through. And it has to be labeled as containing sharps. And that's what they've done here. Okay. As long as you meet those requirements, you don't have to buy an expensive commercial container. You can use an empty laundry detergent container. You can use an empty coffee grounds container. As long as it's labeled, as long as it's hard plastic. Oh, it has to have a lid. I forgot to mention that. It has to be lidded. So of course, if you're going to use this, you'd have to cut an opening into that lid. But yeah, these are used all the time. Again, for sharp things that are not contaminated. A good example in the laboratory, we like to use scalpel blades for things. Sometimes we have to scrape things, again, not contaminated with anything. So we use scalpel blades a lot, and those are going to go into this container. They're sharp, but they didn't touch any culture material. Sometimes we use razor blades in a laboratory, again, as a knife, sort of. And as long as it's not contaminated, we can throw it right into this box. Now, you don't want to use any plastic containers. You won't see plastic containers that are see-through plastic or soft plastic, because that doesn't meet the regulation. And you certainly would never put a sharp item into a regular trash bag, because that's hazardous to the people who handle that trash and throw it away for us. So yeah, it's a heck of a lot cheaper to gather up your sharp things in these containers than to pay for them to be hauled away as though they were hazardous waste. So we separate them. Here's that glass waste container I told you about. This is a cardboard box. You may have seen these in your classrooms. They're tall. They're about 3 feet tall. You buy these again from scientific supply companies. It has a hole in the top. It's a very heavy, thick cardboard. And any glass that, whether it's broken or not broken, but any glass that you just want to throw away goes in here, as long as it doesn't have the potential of having living microbes on it. It would go right in here, and it gets disposed of as trash. So yeah, one of the things that we have to learn about in the micro lab is how we handle our materials safely. And then when we're finished with them, how we dispose of them safely. We have a real responsibility not only to keep ourselves safe, but to keep our colleagues safe, to keep the staff who handles waste materials safe, and to keep the public safe and the environment safe. So one of the things that we learn about while we're working in a micro lab is how to do that and which things go in which containers when we're all done with them. So I have a question from Brianna that says, does it have to have the biohazard sticker as well? If you're going to use a laundry detergent container or you're going to use a coffee grounds container as a regular sharps, non-contaminated sharps, you don't want to put a biohazard sticker on it. As soon as you put that sticker on it, you have said there are contaminated things in here. As soon as you put that sticker on it, you have to autoclave it and then throw it out or pay somebody else to do that. So if it's just an old coffee can that has some old non-contaminated sharp things in it, I can carry that right down to my local recycle center. And I can say, hey, can you get rid of these safely for me? But if there's a biohazard sticker on it, the recycle center will be like, oh, I'm not taking that. You have to call a company and pay to have that taken care of. So that's the difference. Yeah. Yeah, there's a lot of really good, but really complex regulations around how we dispose of things that have a biological hazard associated with them. Body fluids, body tissues, things like that. These are things that are potentially hazardous to people. They may contain pathogens, and so we have to be very careful how we throw them out. Other questions? Okay, so we're going to watch a video together now, and we're going to watch again that list of techniques, how we do those techniques. We're not going to get all the way through the video today, but we'll start into it and we'll see a few of them. And you're just going to want to make sure to, in your notes, that you jot down the things that I'm pointing out in the video as we see these techniques taking place. So I've enlarged this, but I think I'll actually, I think that's probably good. All right, let's go ahead and get started here. When we're working in the microbiology lab, we are often moving bacterial cells from one place to another, from one media to another. And there are different reasons that we do this, but we always have to use our very best aseptic technique while we're transferring cells so that we don't cause contamination to occur. Now, there are lots of reasons why we would take cells out of one culture container and move them to another. One of the most common reasons we do this is because cultures get old. Remember, when we put cells into, for example, a broth culture, there's a certain limited number of nutrient molecules in there. And as the bacterial cells start to grow and divide, they're going to use up those available nutrients. They're also going to be excreting waste materials into that culture tube. And that's going to be dangerous to the survival of the bacterial cells. So from time to time, if we want to keep a culture going, we have to take some cells out of the old culture and move them into fresh media so that they can begin to grow and divide, again, with plenty of nutrients and plenty of space. We call that process subculturing. When you take cells out of an existing culture and you move them into fresh media, We call that subculture.
Whoops, sorry about that.
When we're most common reasons we do this, molecules in there. And as the bacterial cells start to grow and divide, they're going to use up those available nutrients. They're also going to be excreting waste materials into that culture tube. And that's going to be dangerous to the survival of the bacterial cells. So from time to time, if we want to keep a culture going, we have to take some cells out of the old culture and move them into fresh media so that they can begin to grow and divide, again, with plenty of nutrients and plenty of space. We call that process subculturing. When you take cells out of an existing culture and you move them into fresh media, we call that subculturing. Now, as you've seen, there are really two primary types of culture containers that we encounter most often in the laboratory. There are test tubes, and those can have liquid media in them. They can have a semi-solid media in them. They can even have solid agar in them. The other container we see is the Petri dish, the plastic, single-use, lidded dish. We often refer to these as plates. And those agar plates have a thin layer of solid media on the bottom. So those are the two containers that we encounter the most, and those are the two containers that we move cells between most often. So what we'll look at in this video is how to do all of that moving using our very best aseptic technique. Set up in the workspace here, there's an incinerator that's been plugged in and is nice and hot. And there's an inoculating loop. The inoculating loop must be sterile because we would never lay a contaminated inoculating loop on the desktop like this. The only other piece of equipment that I'm going to need to do this demonstration is a Bunsen burner. I don't have access to a Bunsen burner today, so I'm going to use something as a substitute, just for demonstration purposes. And that something is a bottle of hot sauce. That seems appropriate, a good substitute for a Bunsen burner. Now, the next thing I need is a broth tube. And I want you to notice that it's a glass tube. We've talked about how to open up any test tube with culture in it by holding it at an angle and by holding on to the top of the tube in our fingers. What we need to remember is that the inside of a sterile tube of uninoculated broth is still sterile until we place something into it. The outside of the tube, though, once you have touched it, it is no longer sterile. Now, when we're working with a glass test tube, we can take an extra step to protect the sterility inside that nutrient broth tube. And that's to flame the mouth of the test tube when we open it, before we handle it, before we put culture material in, and also flame it again before we close it. Now I'm going to show you what I mean by flame it. This is something we can do using aseptic technique to add an extra layer of protection against contamination. People don't always flame glass test tubes. Sometimes it's just too time-consuming of a procedure, but I want to show it to you because it is, again, an extra layer of protection against accidental contamination. You would never do this with a plastic test tube, though, of course, because you would melt the plastic. So the test tube that I'm holding in my hand has a liquid broth in it, a nutrient media, and bacterial cells growing in it. And I'm going to take a sample of those cells out of this tube, and I'm going to flame the mouth of the tube before and after I remove those cells. So you'll notice that I'm going to take the cap off. I'm going to hold the tube at an angle, just like always. But here's the extra step. I'm going to pass the neck of that tube, the mouth of that tube, through that flame. Let's take a look at that again. Remember, the bottle of hot sauce is my Bunsen burner, and we're pretending there's a flame coming out of that black cap. So I'm going to take the neck, the open part, the mouth, if you will, of that test tube, and I'm simply going to pass it through the flame. Now, what does that achieve for me? The opening to the test tube is the place that's most vulnerable to a contaminating microbe falling in. My hands are up near the mouth of that tube when I'm taking the cap off. And of course, I'm also a source of contaminating microbes, even with gloves on. So, by flaming that opening to the tube, what I'm doing is I'm applying heat directly to the glass. So if there's any microbes sort of right on the lip of that opening, I'm killing those. I'm also briefly warming up the air that's in the mouth of that tube. And by doing that, I actually create a little current, a little swirling current of hot air. And just for a moment or two, that's going to protect the tube. It's going to prevent anything from falling down out of the air and going into the tube. So I've created a few moments of protection from accidental contamination. Now, after I'm finished doing whatever it is I'm going to do with the tube, I'm going to pass it through again before I put the cap back on. Again, if I have accidentally introduced any microbes during my procedure, I just killed them. And I've created a little current of air right at the end, just in case there were any microbes hovering over that area, that opening. I've killed them with that heat. It's a very brief pass through the flame. It's going to kill cells that might be on the lip, the outer lip of that tube that might be a source of contamination. And it's going to create a current of hot air just in the mouth of the tube, just for a couple of seconds, that will keep that entryway free of contaminating microbes. Now we're going to watch a transfer of cells from one tube to another tube. This tube has my bacterial culture in it, okay? And this tube has clean nutrient broth in it. Notice that I put the clean broth tube up higher in my hand. I'm going to widen our view here a little bit. Now, the other thing I'm going to need is a Bunsen burner, and of course I don't have one, so I'm going to substitute in my bottle of hot sauce. Okay, now, first thing I'm going to do is sterilize my inoculating loop. That needs to be into the incinerator until the wire is red hot. For purposes of the demonstration, I'm going to shorten that up a little bit. I'll pretend that we're red hot. Now we have to hold it until it cools off. Remember, approximately 20 seconds. You can count in your head. Now it's important that you get that loop comfortable in your hand for the next part. I've got my tubes at an angle. I'm going to take off both caps. And the reason I do that is because I'm going to be flaming. So I'm going to flame the culture tube first. Now I'm going to take out a sample from the culture tube. I'm going to flame it again. And now I'm ready to close the culture tube. Take one of your caps and put it on. Flame the clean broth tube. Place your sample into your plain broth tube, swirl it around a little just to make sure it all gets in there. Flame it again, close it with a cap. Remember, your loop is dirty right here, so don't forget to put it back into the incinerator to sterilize it once again. If you're working in a laboratory that doesn't use open flame, you can still do a successful broth to broth transfer using your best aseptic technique. You're just going to want to remember to hold both tubes at an angle while they're open. You're going to want to avoid touching the neck or the lip of the tube as much as possible. And you're going to want to close those tubes just as soon as possible to help prevent any accidental contamination. Now let's take a look at the next kind of transfer, which is from a broth tube to an agar plate. Remember, we use plastic single-use Petri dishes in the microbiology lab, and we can pour agar into those dishes and allow it to solidify, giving us a solid media to work with. We refer to those as agar dishes or agar plates. The agar, the solid material, has nutrients added into it so that the bacteria can grow. So this is an agar plate. This happens to have nutrient agar on it. You can just see it. It's a thin sort of beige film on the bottom of the plate. Now I'm taking the lid off just so you can see the inside of the plate. There's just this beigeish sort of sheen to that media. Very, very thin layer of media in here. It feels a lot like gelatin, and it's easy to push your loop into that gelatin. So you have to be careful when you're handling it. Oh, I've mentioned before that we handle agar plates upside down. We put agar plates into the incubator to allow bacterial cells to grow upside down. In other words, we want the agar, that solid media, to be at the very top once it's upside down. And what this does is it allows any condensation that might develop inside that nice warm incubator. It allows it to drip off of the surface of the agar and onto the lid of the plate. This is helpful when we're trying to grow bacteria because that condensation might interfere with the growth of bacterial cells on the agar surface. So we handle them upside down, we open and close them upside down, and we place them into the incubator upside down. I'm going to move my incinerator into place here as I get going. My inoculating loop, my tube full of bacterial culture. Now, this inoculating loop is clean, it's sterile, and I want you to notice that I'm going to start bending this wire. The wire is wire for a reason. When we're inoculating things, especially the surfaces of plates, we can bend this wire to make it easier to spread the cells out. Now you can bend it as far as you want or as little as you want. It really depends on the person handling the loop. But once you bend it into place, you can still get it into the incinerator bent because it's very flexible. It's a wire. So you would heat up the wire just as you always would. See, the bend is still in it. You're going to let it cool. If you have it bent as severely as I do here, once you open up your culture, which I'm doing here, you're just going to want to be very careful as you introduce the loop. Again, you don't want to touch the neck of the tube. Take out some of your culture material. Close your culture. Now you're ready to go. So I'm going to open this dish. Again, it's upside down. And I'm going to keep the plate at an angle so that I can put these cells on the surface. I'm going to place the loop on the surface, and I'm going to make a nice zigzaggy motion all the way across. I'm going to take my loop, remember, incinerate, sterilize, get rid of those cells. So essentially what I've done is I took a bunch of cells out of the culture tube, the broth, and I placed them onto the first place that I hit on that agar. I put a large number of cells, a large amount of cells onto the surface. And then by sliding my loop across the surface back and forth, what I'm doing is I'm spreading those cells across the surface of the plate. So that I'm giving them room to grow on that nice nutrient-rich solid media. Let's take. All right. I'm going to stop us here because we're almost out of time. We'll pick up with that video on Wednesday when we get back together. But does anybody have any questions about anything you've seen so far? Remember, whether or not you're going to flame a glass tube depends on a lot of factors. Number one, can you in the laboratory space you're in? You have to have a Bunsen burner to flame a tube. And a lot of modern laboratories have just gotten rid of Bunsen burners because of the hazard of flaming. I mean, the hazard of an open flame. So if you don't have access to a Bunsen burner, it's okay. You can still do these transfers well without contamination. You just have to remember all your technique, all of your aseptic technique. Keep your tubes at an angle, hold the caps in your hand, don't touch the neck, get your cells in and close the tube as quickly as you can. I want you to learn the flaming technique because it is used in some labs, but it doesn't mean that you have to use it. You can be very successful without flaming glass tubes. All right, I'm going to let you go. Don't forget to do your exam this afternoon at some point or early this evening. Those are due by 8 o'clock. You can jump into your lecture for the week whenever you have a moment. And I will see you again on Wednesday. All right, everybody, have a good afternoon. We'll see you next time.